Oh, am I still here? I'm gonna start the recording. Cool. So welcome everybody to our species distribution modeling for conservation with Wallace workshop. Like I said before, I'm Peter Galanti. I'm here with Mary and Manette. This work is really in collaboration with a lot of other universities around the we would call it the metropolitan area around New York City, Pace University, University of Connecticut, and the City University of New York. And we've been developing Wallace as a tool for conservation with them to make distribution modeling more accessible. Annette, if you could go forward. So our workshop today is going to be broken down into these 4 components, where you first a fairly brief introduction to species distribution modeling, and then talking about how that relates to Wallace, and we're going to be giving some potential applications of distribution models in conservation biology. And then after session 3, we're going to be taking a 5 to 10 minute break. And there'll be time in there for questions. And that's when we'll stop the actual recording. And then section 4 is going to be the rest of the time working with you to walk through a Wallace workflow and create some models. So I think, Manette, if you'd like to take it away.
Yeah. All right, so I'm going to start with an introduction to species distribution modeling. At the heart of species distribution modeling, at the basis of it, is the concept of the ecological niche. And ecological niches can be a little bit confusing for species distribution modeling beginners because there's more than one definition of the ecological niche. Species distribution modeling generally refers to the Grinnellian distribution of a niche, which is very related to the Hutchinsonian definition of an ecological niche. Both of these relate to the environmental and geographic distributions of the species. And most species distribution modeling does not consider the Eltonian niche specifically. So the Grinnellian niche says that there's this set of environmental variables that limit a species' presence in a geographic area due to their physiological effects on the species. And this Gaussian curve on the left shows how given species may respond to an environmental variable, where there's an intolerance zone shown in these 2 red lines on the far outside, beyond which the species can't persist. There is a stress zone between the red and the green lines, or the far and middle lines, where the species can persist but not well. And then there's an optimal zone in the center where the species can thrive. And this environmental gradient can be anything: temperature, precipitation, but this is only one dimension shown in this Gaussian curve. And in reality, this is a lot more of a complex relationship in that there are multiple environmental variables that affect species. So this creates an n-dimensional hypervolume, which is shown on the right, and which is equivalent to the niche of the species. So in the figure of the n-dimensional hypervolume, we might have some temperature on the x-axis, some precipitation on the y-axis, some amount of humidity on the z-axis, and it's n-dimensional because you can add more and more axes to take different types of temperature measurements, precipitation, humidity, all of that. With species distribution models, we try to define the combination of these environmental variables that constitute this hypervolume, And this is referred to, the environmental variables is also referred to as the environmental space or E space on the left. So for example, this is mean temperature on the x-axis and annual precipitation on the y-axis, showing where a species can be. And then try to find the geographic areas in which this combination of environmental variables is present. And that's referred to as the geographic space or the G space, as shown on the right. And favorable environments or climates aren't enough to estimate the distribution of species. So with species distribution modeling, we're actually modeling the realized niche, which is the intersection of a set of environmental variables that enable a species to survive and persist. This is referred to as the fundamental niche and is shown in the diagram as the circle that is labeled A. The set of biotic factors or biotic indicators, which may be where an environment may be favorable, but the presence of a competitor might exclude it from this area. So B is shown on the diagram. The circle of B is the biotic interactions that limit or facilitate the persistence of a species. And then the dispersal ability or the geographic space available for the species dispersal is represented by this circle that's labeled M. So A and B might be favorable, but if dispersal is not sufficient, the species won't be found in a given area. And we can see this back on the previous slide where the geographic space on the right, you have an area that is favorable in black up in the northwest coast of the US and Canada. And you also have this favorable space down in Chile, but few species are actually able to disperse between these two geographic areas. So species distribution models combine known species occurrence records or species presences or presences and absences with these climatic predictor variables, temperature, precipitation, but also topography, soil type, hydrology, and they create a geographic prediction of habitat suitability using some sort of modeling approach. And it's typically a correlational approach like a GLM or MaxEnt, but there are also other options. And through modeling species distribution, and Mary will get into this more later, we can track invasive species and how invasive species are expected to spread across a given area. We can do conservation planning based on occurrences of rare species and where they're expected to be. We can track the migration of zoonotic diseases like Zika virus in response to climate change, and we can produce development and land use policies. All right, now I'm going to turn screen sharing over to Mary for the second part of this.
Thanks, Manette. And if, while I'm getting set up here, if anyone has a very quick question for Manette, you could ask it, but you don't have much time. We will take more questions after my section as well. Are there any burning questions? Seems like not. Okay. I hope everyone can see my screen. Well, so let's talk about Wallace, what you're all here for today. So when we're doing species distribution modeling, there's a range of tools available to do this sort of modeling. On one hand, there are several graphic user interfaces that you can use, and the most widely used example is the MaxEnt GUI, which several of you mentioned. These are very easy to use. They're often called black box. Maybe they're too easy to use. People are very tempted to use the default settings. And it's a bit more difficult to very specifically parameterize your models. The other option is to use more sophisticated computer coding options, which are much more flexible. It's easier to kind of keep up with best practice standards and keep up to date with the cutting edge literature. But there's often a really steep learning curve to be able to implement these computer coding approaches. Wallace attempts to combine the best of both of these options by running R on the back end and using R Shiny on the front end to create a graphical user interface that is powered by R. And we're just highlighting Jamie Cass here, Dr. Jamie Cass, who came up with this idea during his graduate research at the City University of New York. This software was recently published as a software note in its first version, and you can check it out for a lot more details than what we're able to go over today. And we also just wanted to highlight that this is a rather large and growing team. These were the first folks to work on it, but it's now quite a big team, and it's now an international team indeed. And it's a very open community of folks working together to try and prioritize user needs as a part of species distribution modeling tools. So major characteristics of Wallace are that it's free and open source. This is very important to us, and I think this is also very important to conservation users and practitioners. It's meant to be very instructive, so we provide very detailed guidance text and even suggested references for additional reading for each step in the modeling process, such that people can really understand the reasons behind modeling decisions that they're making. The tool is very flexible. The user has several options for each step in the analysis, and it allows you to both download data from public databases or upload your own data at every step. It's interactive. There's an interactive map and different kinds of graphs that do types of sensitivity analysis to help users explore the different steps of the modeling process and make informed decisions. A really great part about Wallace is that the whole point is to help make species distribution modeling more transparent and reproducible. And at the end, as you go through each step, you can download an R Markdown file, which provides the full executable code for the analysis you just ran, so that you can rerun it again easily, or for example, include it as supplemental materials to a publication or a report. And finally, it's built to be expandable. It's built so that people can, because it's open source, they can make their own iteration of Wallace, expand it with their own modules as they wish. And it's also quite easy to expand the base Wallace as new, more sophisticated methods and best practices become available to the community. So Wallace has a modular structure. It's composed of components and modules. And again, this allows for inclusion of new modules quite easily and makes Wallace very easy to expand. Components are the columns that represent major steps in the modeling process, like obtaining occurrence data, processing it, etc. And then underneath that, the blue diamonds are modules that represent different specific tools that are available within each of the components. Oops. So now I'm going to go over some of the potential applications of species distribution models in conservation biology and to inform conservation activities. A common application is to estimate potential changes in range distributions after climate change, or areas of a species range that might be the most vulnerable to climate changes. In this example, it's showing you chytrid fungus, which is a severe amphibian disease responsible for massive die-offs. And in the figure, you can see some dead frogs in the Andes. Unfortunately, it's a very graphic image. In this study, they applied species distribution models to predict the distribution of chytrid fungus under different scenarios of climate change and how that might affect risks of extinction for amphibians in the future. Another example is something that we're working on with Beth Gerstner, who's now a PhD student at Michigan State. Here, she was interested in evaluating the status of a rare and newly described threatened species. This is the olinguito, which is a carnivore endemic to the Andes. It's strictly associated with forests, but it has only been described relatively recently, but with museum specimen information. So it has very few occurrence records available, and most of those occurrence records are for years where we don't have high resolution forest cover data available for them. So we might not really be able to interpret very well a species distribution model that is created with climate only when we know that they're strictly tied to forest. It is possible to include forest in the training of the model, which is an option for many people and many different species. But in this case, when most of the data occurrences are from historical years where we don't have forest data, that might not be appropriate. So in that case, an option is to take a climate-based model and then create a mask that takes into account forest cover using a data-driven approach where you might create a threshold for forest cover based on whatever small set of more recent occurrences that you do have that could match up to forest cover. And then you could mask out the recently deforested areas from your climate-based distribution model. We're working on some R packages to add to Wallace that would make this a part of the workflow that we're showing you today. And we hope to, in addition to this, take some additional steps to calculate different IUCN metrics that are of interest, like extent of occurrence, an area of occupancy. At least the upper bounds can potentially be informed by species distribution models. Another very common example of using SDMs in conservation is for trying to predict the risk of invasive species spread. This is another example from a collaborator, from Matt Aiello-Lamans, who's a Wallace developer as well. This is of the glossy buckthorn, which is an invasive species in the United States. And Matt used species distribution models combined with demographic models to predict the historical spread of this tree in the US. So he was able to take time steps and account for population growth and dispersal in predictions of how this invasive species was move across the US. And because he was looking at historical spread, he could validate that with data and found that it worked quite well. I think these are some really interesting examples for you guys. And we're gonna take a break in a moment, but I just wanted to say, in any application of species distribution models to conservation, it's important to have a good sense of the uncertainty of the models that we're working with. And Wallace does a good job of helping to explain that in the user guidance, and we're gonna try and talk about it as well. Because understanding that uncertainty really helps to communicate whether your model is more of a trend versus a prediction, and how to make models that can be informative, but aren't necessarily telling practitioners and decision makers, oh, this model is 100% right, because that's probably not true. But we can do the best that we can to account for known biases in data, and we can do the best that we can to validate the models that we do have. So we hope to show you how to do that today, and we're really looking forward to your questions and also talking more about your specific cases that you're interested in applying species distribution modeling to conservation, and how we can walk through you know, any specific issues you're thinking about regarding uncertainty and validation and the nitty-gritty of these models. So I think we're going to take a moment now to answer any questions you have. And after a little question and answer discussion, we will take a break, and then we'll begin the software walkthrough with all of you, which we're excited about. And I'm gonna leave this slide up, which I think, can you still see it? And
Yeah,
this
we still
has,
see it.
good. So this still has the, here we go, sorry. This has some links here for you to check out some tutorials. There's also a YouTube channel for Wallace where we have various webinars and presentations that we've given before. And you'll have access to that. And on the Wallace website on GitHub, there's also a link to our Google group. And so we will try and answer all of your questions today about the software, but if you begin teaching with the software, if you begin using it in the future and you're having issues, the Google group is a really great place to go for any questions. So yeah, if anyone has questions for me or Manette or Pete before we take a quick break, you can pop that in the chat box. Or you can just unmute yourself. I think there are few enough of us that wouldn't be a problem. Any questions?
I have one, and you may be about to cover this, but
Sure.
does Wallace have a special kind of module for aquatic systems, like along stream networks, as opposed to terrestrial?
Great question. We don't have a specific module for it, but we have some users, some freshwater focused users that we've been working with as we are expanding Wallace to do more conservation related things like the IUCN functions I was talking about. And we're working with the freshwater fish group to flesh out a full use case to make sure it works. But as of right now, because their users can upload their own data sets, it should be totally feasible to use Wallace to model freshwater species. So if you have a stream network shapefile, you with some assistance with pre-processing, I don't think it should be an issue. You should definitely be able to use it. And we'd love to have a vignette or a user guide that would be more specific to that, because I know there are probably other more specific freshwater concerns. And that is something that we're working on. So yeah, Pete, did I answer that sufficiently? No.
Yeah. Basically, nobody on our team works with freshwater systems, so we haven't had the ability to add in that kind of module yet.
Yeah, but it should work now. We've had some folks do marine species and coastal species. We had a PhD student at CUNY who did a marine species of starfish, and it worked. She was surprised how well it worked. She actually was able to go out and ground truth and survey in one of the predicted areas to find the species, and it was there. This is Molina I'm talking about. Yeah, so.
I'm putting the Wallace YouTube in the chat right now. Oh, somebody already did it. Perfect.
Cool.
Both videos that we have on there.
I have a question.
Yes.
So from the beginning, you basically, so it seems that the species distribution is really meant for more environmental niche, but not Eltonian niches. But I can also see it, or maybe I've seen it apply using biodiversity metrics, or maybe presences of multiple species at the same times, and therefore like try to get at the Eltonian niche, or is it more that is, or is it a better advice to don't get into that unless you have like very good ecological information of trophic level or some sort? I guess, let me simplify that question. Like, is it that you can use this kind of model for an Eltonian niche to infer into the Eltonian niche of a species, or you will suggest to kind of not try to go that way?
You want me to take this one? Okay. There are a couple of different directions you can go to infer a more Eltonian niche. In terms of ways to use Wallace specifically, and then I'll build out. One way in which a number of people get at Eltonian niches through species distribution models is you model the more Hutchinsonian Grinnellian niche of each species in particular that you're interested in, and then you plot the overlap between the species. And so in terms of biodiversity, species richness in an area, if you're interested, for example, in like the species richness of, I believe you said your study species was oaks in Southern California. So if you're interested in the species richness of oaks, you could create species distribution models for each oak species and then layer them on top of each other and add up for each grid cell, whatever size that is, add up the number of oak species for which the climate or the area is suitable. So that would be the biodiversity side. If you're looking at, for example, an interaction between Southern California oak species and I think in terms of seed dispersal, because I do seed dispersal work, so the birds that disperse acorns, then you would do something like create distribution models for all of the oak species that you're interested in, distribution models for all of the bird species that you're interested in, and then layer them on top of each other. Or if you were interested in how well the oak species predict the bird species, you could do something similar and then ground truth that. There are ways to combine species distribution models of multiple species to specifically get at interactions. Those are for those you're getting into things beyond at least what I know Wallace can handle, and you're getting into, and Mary and Pete can correct me if I'm wrong about this, but you're getting into programming your own species distribution models and your own you know, depending on how you do it, for example, linear correlational relationships where you're using the species distribution model of one species and the species distribution model of the other species in conjunction to make this giant species distribution model. That's more complex, but there are a number of papers about that, and it is a growing body of research within the field. Did that fully answer your question?
Yeah, no, that was great. Thank you.
You're
Very
welcome.
nice.
Mary and Pete, anything to add that I might have missed?
That was great. I mean, the only thing I'd add is that Minette is an expert in this, and she's underselling herself because she has done joint species distribution modeling for her PhD before her postdoc with a very sophisticated hierarchical Bayesian framework. So it's a little beyond what we're doing in Wallace right now, but we are thinking about, we have plans to actively in the next year actually add to Wallace a couple of things that get at what you're talking about. So one thing we're adding to Wallace is a feature with, so it's not in what we're showing you today, sorry, but we're doing it the next year. It's a feature with support vector machines that would help you mask out parapatric species that you know is competing with your species of interest. And that's, you know, common when there's active competition happening. And in that case, the species distribution model using abiotic factors is always going to overpredict and not take that interaction into account. So we do have something where you could mask it out after the fact instead of including it as a variable before. You could in Wallace create a model for one species and then try to include it as a predictor variable and model the next species. So that is something you could do in Wallace. People have tried. And the only other thing I'd add is the thing about calculating like species richness or species diversity and stacking the rasters after the fact. We're also trying to add a component to Wallace where you could do that. So we're currently showing you Wallace version 1+, and one of the biggest advances of Wallace version 2 will be the ability to handle multiple species at the same time. And we'll also be including some analyses where you can compare the niches of two species and do like a niche overlap analysis, as well as later on calculating species diversity and stuff. So these are things obviously a lot of us are interested in and want to do, and so we're working on that actively for the next version.
And even though it's not in the functionality of Wallace right now to automatically stack it, it is incredibly easy in R to take the raster outputs and stack them.
I think if there are no other questions, I think the plan is to take a short break now, just like a bathroom break, get some